Hello and welcome everybody. Thank you all for joining our virtual COVID-19 panel discussion. My name is Emily Begain. I am research faculty in the Intertribal Research and Resource Center at United Tribes Technical College, and I'll be moderating our discussion tonight on behalf of the Visiting Scientist Series here in Bismarck. The Visiting Scientist Series is a partnership of academic and professional entities that come together to bring STEM, science, technology, engineering, and math to the community. And we are very excited for this opportunity to feature regional scientists and have them share their expertise with us. Our panelists tonight are Dr. James Peliska, Dr. Catherine Brissette, and Dr. Joshua Wynne. Thank you all panelists for taking time to share your knowledge with us. This evening, each panelist will give a 10 to 15-minute presentation on their topic, and then we will open the floor for questions. Attendees' audio and video is disabled. However, questions can be entered into the Q&A feature. It looks like a comment balloon with a question mark for the question and answer session. We may not get through all of the questions, but we will try our best to answer as many as we can. I am asking everyone to please leave politics on the side tonight and focus on the science. I'm looking forward to a great discussion, so let's go ahead and get started. I would like to welcome our first panelist, Dr. James Peliska. Dr. Peliska received his undergraduate degrees in biology and chemistry from Northland College in Wisconsin. He graduated from the University of Wisconsin-Madison with a PhD in organic chemistry. He was awarded a National Institutes of Health postdoctoral fellowship at Penn State University, where he started his current research on HIV drug discovery. He began his teaching career at the University of Michigan School of Medicine and has been with the University of Mary for the past several years as a professor of biology and the administrative chair of sciences and mathematics. Dr. Peliska will be talking about viral zoonotic diseases and their public health ramifications. So Dr. Peliska, the floor is yours.
Thank you. I'm going to pull up a PowerPoint here, hopefully. And can you see the PowerPoint?
Yes, we can.
Okay. So I have the honor of starting off this session. I want to thank the organizers of the Visiting Scientist Series for inviting me this evening. I've been studying virology in mostly biochemical ways over the last 20-some years. This is a great opportunity for me to talk about something that's very current and important. And the first slide here is kind of the state of the COVID pandemic as of about 2 o'clock last night when I prepared this slide. And I just show you this as a prelude to what I'm going to talk about. My goal is within the next 10 to 15 minutes is to give you a very, very brief, very overview of how we could have possibly gotten into this state. How is it that this pandemic could have arisen from what seems to be a single location on the other side of the world? So I want to start off, since I know we have a broad audience of individuals here perhaps viewing this, I want to keep it as general as possible while introducing some technical terminology. So I think it's best if we perhaps start with a few definitions before we move forward. So since the etiologic agent of this pandemic is a virus, I wanted to first put up a simplified view of a viral replication cycle and highlight a couple of aspects that are important for not only my discussion, but perhaps a number of the questions moving forward. In that viruses really aren't considered living organisms because they don't replicate autonomously or have their own metabolism. They have a requirement to infect a living cell, which we call the host cell. And as such, we tend to call viruses infectious particles rather than organisms. And we talk about active or inactive viruses rather than living or dead viruses. So that might come into play if we get around to talking about perhaps strategies with vaccines and so on. So here's a few definitions that I think might help us as we move through all of the talks and perhaps into the Q&A. And I just want to go through some quick definitions. Some of these are common in their language these days, but I just wanted to go through them. Zoonotic disease is an infectious disease caused by some pathogen, In our case, it's a virus that crosses a species barrier specifically from animals to human. And many of these zoonotic diseases are what we term emerging infectious diseases, and these are defined by outbreaks of previously unknown diseases that are increasing in incidence in various locations around the globe over the last 2 or 3 decades, and then persistent infections. And I just listed a few of them there that many people will be familiar with because they've been in the news over the last 20 years: HIV, Lyme disease, SARS, which is the one we're dealing with now, a strain of SARS, dengue and West Nile disease, and Zika, which was a big issue when I was down in Florida several years ago. And another term is natural reservoir, and these are populations of animals in which an infectious agent normally resides. So it could be a bat population, it could be a chimpanzee population, it could be some kind of population which normally contains the infectious agent. Now, these infectious agents don't have a pronounced effect on the health of their primary host, and that's how they're able to propagate and reside within these natural reservoirs. And then for our purposes today, one of the biggest definitions is what's called the spillover event. And a spillover event is that event in which a pathogen, which is prevalent in some natural reservoir, jumps or is transmitted or spills over into another population. In our case, it's going to be humans. And of course, COVID-19, I just want to define for those of out here, because sometimes this term is misused. COVID-19 is used to define the disease state in the pandemic, or coronavirus disease 2019, and SARS-CoV-2 is actually the etiologic agent or the causative agent of the disease COVID-19. And of course, it gets its name from the spikes on the surface of the virus, which make it look like a crown. Okay, so as far as spillover, let's kind of dwell on the spillover event for a couple minutes. And what is a spillover? Well, it turns out that spillovers are very, very common in nature. But not all spillover events can lead to pandemics, or do lead to pandemics, thankfully. So almost 2/3 of all human viruses are zoonotic in nature. In other words, they come from a non-human animal population. And oftentimes, as you can see with rabies, you'll find that you have a spillover event. In other words, the rabid dog might infect a human host, but that human host, which is now infected with rabies, can't transmit that pathogen to other humans. So in other words, that host is considered what we call a dead-end host, and the infection stops with that individual. And many of the human viruses of zoonotic in nature are like this. Some, however, spill over, such as Ebola and SARS, spill over into human populations, and then they remain infectious agents that can be passed from human to human within the population. This is a distinctly different problem and possibility from the first case, which can lead to many more problems for global health as we are living out, as we see before us today. So now, this passing on from human to human, this transmission, can be very efficient and prolonged, or it can be short and only go through a couple cycles. Sometimes the transmission from human to human is quite readily able to transmit, such as what we're experiencing with the COVID virus. Sometimes it's less easily transmitted, such as with Ebola virus. So Ebola virus requires usually contact with blood for the passage of the virus from person to person. And then finally, we have some spillover events that have since adapted. The infectious cycle has adapted so that there is no, the original host give rise to a new stable reservoir. In our case of our discussion today, you can consider that humans in the case of, for example, HIV-1. So HIV-1 had its origins in the chimpanzee and ape populations in Africa, but is now stably transmitted really solely through human-to-human transmission. So that's also a distinctly different sort of scenario than the one we're dealing with the SARS right now. So I want to focus my attention on this slide, and this slide presents the stages associated with the development of a zoonotic pandemic. So it's really important that we understand these stages, not only to understand the pandemic situation that we're in today, but to also prevent future pandemics from occurring. And so far, we have not been able to do a good job of predicting when a pandemic will arise. So we really have to take note of these different stages and then try to develop better methods of preventing the progression of these stages. So let me just briefly go through these. In stage 1 is what's called the pre-emergent state. And in there, you can see on the bottom there, in this state, the animal reservoirs are exchanging the pathogen, in our case, a virus, between themselves. And then some ecological event or socio event occurs, such as large-scale environmental or agricultural changes or demographic shifts, moving livestock into regions that weren't involved in livestock before, transporting of wildlife for food to various locations. Some event occurs that allows for the virus that is in its normal natural reservoir to jump or to spill over into these secondary hosts. And you can see, for example, the cow or the pig or the sheep over on the right-hand side. This is still all considered phase or stage 1 because there's no humans involved yet. And I want you to notice when that contact with humans is an essential part, of course, of this emergence process, because in the second phase, in stage 2, is what we call localized emergence. And the drivers for that is when there's a spillover from livestock or wildlife population coming in contact with humans through the butchering of wildlife or fomite exposure to the exposure of the virus on fomites, or food markets and livestock farms and so on, a spillover occurs from either the wildlife itself or these secondary hosts. And you can see in the middle there, the green represents small-scale infection and recovery from these various infections that might occur, and the red ones indicate spikes in infection due to exposure to these viruses from these sources. Now, this is the first stage in which humans are now involved, and now the virus has transmitted to the human, and now comes the time, but if you consider back in the previous slide, is whether or not human-to-human transmission is possible, or whether or not we have the human infection represents a dead-end host. If it's a human-human transmission, the next scale up is stage 3, which would be the full pandemic emergence, and that's where we have person-to-person spread. We begin to see large-scale spread, and then that's exacerbated by the incidence of now global travel and the international movement of various reservoirs of animals through trade and individuals moving around the globe, particularly through air travel. So if we look at this slide here, this is the air traffic around the globe, and you can see quite easily how a pandemic such as the COVID-19 could spread readily across the globe. So we have to begin to understand the rules or the drivers for these different transitions from a pathogen that's located within a natural reservoir, spilling over into secondary hosts, into eventually the human population, and then how that, in this case, virus can spread from human to human, and then how it can spread globally. So this shouldn't be a big surprise to anybody that this pandemic is occurring. It has seemed to be only a matter of time, but we have to figure out what these rules of emergence are and do a better job at being able to predict and halt the progression of this emergence cycle, because this is almost certainly going to rear its head again in another pandemic. So this is going to require a interdisciplinary effort, and I tried to write down as many of the rules and kind of sectors of science and other aspects, sociology and politics and business, that have to come together in an interdisciplinary effort to try and understand these drivers, and also to try and find ways of surveilling and implementing procedures to halt this emergence progression at the first stage, right? The first stage is the natural stage of how the virus moves within its own natural reservoir. Our goal is to prevent it from progressing from stage 1 to stage 2 and certainly to stage 3. And that's going to require our understanding of ecological, virological, pathological, socioeconomic, political, and economic rules associated with these driving forces. And you can see on the right hand there's a large sector of different individuals, disciplines that are going to have to be involved in this. Because this pandemic that we're experiencing now is by no means probably going to be our last if we don't do a better job at this, because here is a short list of zoonotic diseases from the World Health Organization's emerging infectious disease list. And you can see in red are viruses, purple bacterial, green are parasitic, and black are prions. And the majority of human pathogens, about 61% of known human pathogens, are zoonotic in nature. And you can see there, the list is quite extensive. And there's certainly new ones out there already, because as I said, this spillover is quite common. And we have to learn what the rules are and the driving forces for these spillovers, and hopefully glean what we get from this pandemic and work hard using all of the partners involved to try and prevent the next one from occurring. So with that, I have other slides, but that are available that I can pull up later on in the Q&A that involve more of the virological and molecular aspects of this virus in the replication cycle, that perhaps if I have a question, we can pull up later. So that's my presentation for now, and I look forward to hearing the other speakers, and I look forward to answering as many questions as I can at the end. So thank you.
Thank you very much. Great. Okay. Our second panelist will be Dr. Catherine Brissette. She received her Bachelor of Science in Zoology. Is there a bunch of feedback? How about now? Is that better? Okay. Let me start again. Dr. Catherine Brissette received her Bachelor of Science in Zoology from Louisiana State University. She completed her Master of Science at the University of Vermont and received her PhD from the University of Washington, studying oral spirochetes, which are spiral-shaped bacteria. She then pursued a postdoctoral fellowship at the University of Kentucky, studying Lyme disease and the associated spirochetes. And her research on Lyme disease continues in her current position in microbiology and immunology at the University of North Dakota as part of the Biomedical Sciences Department. Dr. Brissette tonight will be talking about her work with COVID-19 test sample transport medium. So Dr. Brissette, thank you, and the floor is yours.
All right, how about now? No luck? All right, well then I'll just talk. Dr. Peliska gave us a really nice overview of the coronavirus and also emerging infectious diseases and spillover events. And if anyone is really interested in reading more about that, I can highly recommend a book called Spillover It's available on Amazon. It reads like a mystery thriller and really delves into how these events happen. So as Emily mentioned, I study Lyme disease, so I work on something completely different. But where I come into this story was back in the early days of the pandemic. There were a lot of shortages, and you probably saw a lot of these things on the news. There were shortages of masks, there were shortages of gloves, there were shortages of the special swabs that are needed to take the samples. And there was also a shortage in something called viral transport media. And what this is just kind of a broth. And if any of you have been to some of these mass testing events and had either the nasopharyngeal swab or the one in your back of your throat, both awful, I've had them both. Then they take that swab and they put it in this little tube full of viral transport media. And this is just something that helps stabilize that sample until it can get to the lab and we can test for the presence of the virus. And in the early days of the pandemic, this viral transport media was in really short supply. Now, this is something you don't just use for testing for COVID. This is something that's also used for testing for influenza and other respiratory viruses. So Mary Jo Royko, the microbiology program director of the Department of Pathology and Lab Sciences at Altru Hospital in Grand Forks, had reached out to the School of Medicine and said, hey, we can't get any of this viral transport media and we need to do a bunch of sample testing. What are we going to do? Is there anybody there who could potentially help us? And so two of us, myself and Dr. Matt Nilles, said, sure, we can help with making this viral transport media. So again, I'm a microbiologist. I study bacteria. So does Dr. Nilles. He actually studies the bacteria that causes plague. But if there's one thing that microbiologists are really good at, we're really good at making media. We're really good at making the broth and the things to grow and stabilize both bacteria and viruses as well as human cells. And we're also really good at aseptic technique. So we're really good at keeping things really clean and really sterile. So this was a multi-step process. So the first thing we needed were little tiny glass beads. So inside each of these tubes of viral transport media, there's these little tiny glass beads, and they're about 3 millimeters. So that would probably be smaller than like my little earring stud. So these really tiny glass beads. And they're inside the tubes of this viral transport media because they help break up all the secretions that are present. You can imagine if you get a nasal swab or a back of the throat swab, there's going to be a lot of mucus and saliva in there. So these little glass beads help break everything up. Now the problem is we could get these glass beads and somehow we've got to get those glass beads sterile and clean and into the right proper tubes. And we needed precisely 3 beads in each tube. And this is because the FDA and the CDC had very strict guidelines about what goes into making this viral transport media. So my colleague, Dr. Nilles, figured out a way to get these beads and get them into these little tubes. So that was the first step. The second step was to make the actual media. And it's like cooking. You have a recipe, you gather the ingredients, you mix things together, and then you have to sterilize this. And we do this in an autoclave. And this is basically like a pressure cooker. If you're familiar with canning, you know that if you're canning something really acidic, like tomatoes, you can just do them in a boiling water bath. But if you're doing something like green beans and you're worried about botulinum toxin, you need a much higher temperature in order to kill the bacteria that make that toxin. But the only way you can get that high temperature without all your water boiling off is to do it in a pressure cooker. And that's basically what an autoclave is. So Matt would painstakingly put these 3 little glass beads in these little tubes, sterilize those, and then send a message out. And then Mary Jo would come into the lab, because we're all trying to keep physically distant. She'd come in and she'd make this media, and then she would put that in the autoclave and sterilize it. And then I would get a message, and then I would come into the lab and then take that media and the tubes and combine them together in the special little plastic tubes. So not a very complicated process, but you think about the companies that would normally make this, they have all kinds of automation, and we're doing this all by hand. But I have to say that in the early days of the pandemic when we didn't know a lot about what was going on, and there was a lot of stress. And I know that many of you probably experienced, you're trying to work from home, or maybe you're not able to go to work because your business is shut down and you've got children at home doing distance learning. It's all very, very stressful. It was really helpful for me to have something hands-on that I could do and that I could contribute. So the final stage of this was that we had to make sure that the media that we made met the specifications that the hospital needed. And so the North Dakota Department of Health did some quality assurance on our media and made sure that that it was sterile, that it was clean, and that they could spike it with SARS-CoV-2 and be able to run the tests that they needed to run and show that virus was in there. So that was my little part in helping out with the beginning of the pandemic. Altogether, we made about 9 liters of viral transport media, and that comes out to about 3,000 tubes and 3,000 tests. So that was our little contribution to the cause. So I will stop there.
That is great, Dr. Brissette. Thank you so much for sharing that information and that process with us. Okay, our final panelist for the evening will be Dr. Joshua Wynne. Dr. Wynne received his Bachelor of Arts degree and medical doctor degree from Boston University and Boston University School of Medicine, respectively. He completed an internal medicine residency and a cardiology fellowship at Peter Bent Brigham Hospital, which is part of Harvard Medical School. Dr. Wynne earned a Master of Business Administration degree from the University of Chicago and a Master of Public Health degree from the University of Michigan. Currently, he is the Vice President for Health Affairs and Dean of the University of North Dakota School of Medicine and Health Sciences. Dr. Wynne served as interim president of the University of North Dakota from June of 2019 to June of 2020. He's also a practicing cardiologist with Stanford in Fargo and East Grand Forks. And most recently, Dr. Wynne was appointed as Chief Health Strategist for North Dakota. Tonight, Dr. Wynne will be talking about the clinical features of SARS-CoV-2 and possible control strategies, Dr. Wynne, thank you, and the floor is yours.
Thank you very much. Nice to see everyone. I'm glad I didn't have any slides because I probably wouldn't have been able to share them with you. So good to see all of you, if only virtually. So I thought I would just chat a little bit about the pandemic, especially as it affects North Dakota, and just give you some observations and some thoughts about where we're headed from here. Now, from a clinical standpoint, the important thing with any sort of infection that affects a lot of the population, there are 2 major factors. The first factor is how infectious is the agent, and the second is how virulent is it? How much damage does it cause? So in the first one of how infectious it is, there are various ways that we look at this clinically, but let me just compare it with a couple of other common viral diseases, because SARS-CoV-2 is sort of in the middle. Now, there are really, really infectious bugs like measles. When one person gets measles, they can spread it to many, many other people. On the other hand, influenza, H1N1 being a recent version of it, is sort of modestly infectious. And SARS-CoV-2 is closer to influenza than it is in measles. So it isn't highly contagious, but it isn't nothing either. But where there's a really big difference is the impact that it has on the population. Influenza over the years typically has a mortality of somewhere around 0.1%. SARS-CoV-2 in North Dakota has a mortality over 1%, so it's 10-fold higher. The other difference that we see with the current pandemic is that most of the morbidity and mortality is in the elderly. With many other diseases, we see a bimodal distribution where the young are at risk and the elderly. Not this one. This one is mainly the elderly. So if you look at people who have passed away, as well as the people who are in the hospital, it goes up as age goes up. Now, that does not mean that the young are immune or don't get sick. We've had mortality in North Dakota of younger people, but it's pretty rare. One of the things that we've seen recently is that the age distribution of people who are getting the pandemic virus is actually getting younger. The 20 to 29 year of age group is the most common group now that's getting the disease, and it actually goes up from there, that the next couple of decades actually have a lot of disease as well. As a university professor, given that 20 to 29-year-old prevalence of disease, we're really concerned about on the college campus. And what we're seeing is that a lot of the spread, especially in the Midwest, appears to be coming from small group settings, not the large things, not the Sturgis rally, not that sort of thing. It's more small gatherings. So unfortunately, think about that as we have Thanksgiving coming up, because I am really concerned that as many of us get together with families, even in small settings or small groups, that we may have the potential for a lot of spread of the disease. As a matter of fact, the University of North Dakota has recommended to our students that they actually shelter in place, that they stay on campus for Thanksgiving rather than going back home. If they do choose to leave, and obviously there's a lot of pressure why we all want to be with our families, then we're recommending that students get tested before they leave and as soon as they come back on campus. And that way we hope we can limit the transmissibility of the disease. Now, let's talk about the importance of testing in general. The standard test that's used is called a PCR test, and I think Dr. Brissette talked a little bit about the swab and so forth, but the testing of the disease itself is exquisitely sensitive to the bug, but it takes a lot of technology to get that result. The DNA, the code, the genetic code, not DNA, but the genetic code has to be multiplied multiple times so that you can detect it. But when you do, it's very reliable that the person actually has the SARS-CoV-2 virus. One of the things that it doesn't necessarily tell us is how much virus load that person has if we just hear they have a positive test, or whether they're still infectious. So we know that there are people who will be PCR positive for days or even weeks after they have the infection, but that doesn't mean necessarily that they may be able to transmit it. Now, newer tests are coming online now. The federal government has invested a lot of time, effort, and money in newer tests that don't look at the genetic code as in PCR testing, but actually look at specific proteins that make up the virus, and these are called antigen tests. And one of the ones that you may have heard of is called Binax Now, which actually is becoming available in North Dakota. The advantages of this test is that it's point of care. That is, you don't have to send a sample to Dr. Brissette's laboratory. It's fast. It takes only about 15 minutes, and it's much cheaper. It costs about $5 compared with $125 or something for the PCR test. It's certainly not as reliable though, especially in people who don't have symptoms. But it still increases our ability to identify people who are capable of transmitting the virus. Why is that important? Because the key to limiting this pandemic is to limit the chance that someone has to pass it on to someone else. That's the way this virus is transmitted, in droplets and so forth that come out of your mouth or out of your nose and gets transmitted to other people. We don't know how much occurs because the virus gets on surfaces. That's probably not a major way that people get infected. And it's still uncertain how much there's what we call aerosolization. That is the virus getting into the air and remaining in the air for an uncertain length of time. But we know for sure that droplets will transmit the disease. So what do we do to try to limit the pandemic? There are really 4 things that you can do and that I can do. 1 is wash your hands. 2 is wear a mask. 3 is watch your distance. That is how close you are to other people. And 4 is keep gatherings to the least number of people possible. The best number of people in a gathering is 1. That is to stay away from everyone else. That's the best way to protect yourself. But, life has to go on. So trying to do those 4 things are really the main way you and I can try to limit the pandemic. Because up until now, we've tried the standard approaches, which is identify people who are positive, identify their contacts, and quarantine their contacts. But in North Dakota, we've reached the phase of the disease now that those measures will not be enough. The way we can reduce the number of people getting infected and the number of people in the hospital is through our behavior. And our behavior is to do what 4 things? Wash your hands, wear a mask, watch your distance, and limit the number of contacts. And that's what we need to do now over the next several weeks to try to turn this around, because otherwise hospital capacity is going to be really strained, and we're going to start looking like New York City, which is something I don't think we want to do. Finally, how will the pandemic end? Well, there are really only 4 conceivable ways that this is going to end. And I would suggest that there is only one good one. The first is that the virus mutates in some way so it becomes less virulent and less infective. Infectious. There's been no evidence that it does that. It mutates, but it doesn't do it in those ways. The second is that we get better treatment for the infection, and there have been some encouraging ones. Just recently, this week, monoclonal antibody therapy has been approved for outpatients who have mild to moderate COVID. Dexamethasone also has had some benefit, as has remdesivir. So there are some treatments out there, but they're not game changers yet. The third one is the one we don't want, which is called herd immunity. Herd immunity means when enough people in our community get the disease, that the disease stops its transmission. The trouble is that with this bug, we probably need 60 to 70% of the population infected to get herd immunity. Remember, I said the mortality is over 1%. So that means we're going to have a lot more deaths if we wait for herd immunity. Rather, what we want is the 4th way of ending this pandemic, which is to get to herd immunity, but through a vaccine. You all heard the encouraging results about the Pfizer vaccine that has very encouraging results. There are 5 or 6 vaccines that are making their way through the evaluation process, and we are hopeful that one or more vaccines will be available maybe by the end of this calendar year. So I usually tell people that a reasonable expectation is that 2021 will start looking better, especially as we get into the second, third, and fourth quarters of 2021, as natural immunity increases a little bit, but hopefully vaccines come on a station and make a major change. So I think I'll stop at that point, and I'll be happy to try to answer any questions. Thank you.
Thank you very much, Dr. Wynne. That was great. Okay, I think we will go ahead and open the floor for questions. And I believe, Dr. Maskey, you will be moderating. Okay, so yes, you guys are all welcome to post questions in the Q&A section, and we will go ahead and ask those to our panelists. I can start with one. So why are there such a variety of symptoms, and why is each individual's response to this virus different?
Is that for me or?
For any of you that would like to contribute.
Well, I mean, as far as the symptoms, you know, we know even with sort of the common cold or the flu, people may have different manifestations. But, you know, common manifestations of people who are symptomatic may include fever, may include headache. One of the unique ones is loss of taste or smell. That's a real tip-off in this. People may have cough, reflecting the lung involvement that people can get, sore throat, and so forth. One of the interesting things I'd like to stress is a lot of people seem to get the disease and are either asymptomatic or pre-symptomatic. Asymptomatic obviously means they don't have symptoms, but pre-symptomatic means that they have the virus, it's replicating in their body, but they haven't developed symptoms yet. And some studies have suggested that that's actually when patients may be most infectious. So they haven't developed the symptoms. They're going to, but that's a day or two later. So that is one of the reasons why my assumption when I'm getting into any social encounter, I assume that the other person has COVID. You know, that's, I'm saying that slightly tongue-in-cheek because that sounds like I'm paranoid. But with this disease, I actually am paranoid, and it's for that reason that you can't tell just by looking at someone or the absence of symptoms that they're not that they're not infected. So, you know, some establishments check temperature, and I think that's helpful. But it may be that the person does not have a fever and yet can transmit the disease. Why do people react differently? I wish I knew. There was some thought that some patients get a very aggressive reaction of their internal protective mechanisms, a so-called cytokine storm, but some recent data where they actually measured levels suggested maybe that isn't the case. So we're not sure why it varies. The one thing we do know is that the patients who are most likely to have significant symptoms, especially lung involvement, are people who are older or have underlying conditions. And the underlying conditions can be as relatively non-threatening as high blood pressure and so forth. So this is, again, an argument why natural herd immunity is a bad idea, because a lot of us walking around who are otherwise appear to be healthy may have a condition that could predispose to major morbidity or mortality.
Thank you. The next question we have is from the audience: should we be testing once a week if possible?
Does anyone else want to answer? I don't want to soliloquize here.
Well, I don't know about frequency, but that frequency, perhaps Dr. Wynne would be better equipped to answer that. But I just wanted to kind of piggyback on what he was saying, that to get from an epidemiological perspective, to get a handle on where we are, testing is absolutely essential for the very reasons that Dr. Wynne was mentioning. The long incubation period of the virus, this ability to have the virus and be asymptomatic, basically the zero days between symptom onset and maximum infectivity. Again, if sickness is indicated in a patient, you usually quarantine them or you hospitalize them depending on the severity. But if you're at the time asymptomatic but is still infectious, this is contributing to the rapid and pronounced spread of this virus. So, and then again, as he mentioned, this high proportion of those who are infected but have mild or no illness, this again allows for undetected transmission. So all this together really highlights the need for testing to really get a handle on who is infected and who is not, because not everybody is going to be displayed, right? So, just to comment on another one about how infectious this is, from a virological perspective, from a biochemical perspective, there's some unique aspects of this virus that even differ than the SARS-CoV one that was in the early 2000s, in that the protein that binds to the receptor on the surfaces of the cells, particularly in the lung and esophagus, and respiratory tract, are very much more tightly binding than in the CoV-2 than in the original one, which is making it much more efficient at infecting. At least that's the theory. Much more efficient in infecting the cells. So. All these factors together, I think is certainly testing is one way in which we're going to get a handle on this, so that people know when they have to isolate and quarantine, as opposed to just assuming that they're not sick because they're asymptomatic.
Okay, I'd like to jump in there about the testing. So, to sort of illustrate, there's a window when you could get tested and not have a positive test. So for instance, I was working very closely with my graduate student within 6 feet. We were masked, but that afternoon, after we'd spent about 2 hours working in close contact with each other, she got a notification that she was a close contact of someone who was COVID positive. So she went into quarantine, and then a few days later she started getting symptoms, and so I quarantined myself. I went to the first testing event that was available, and I was negative, but it was within like 5 days of that exposure. And so even if I had COVID, that test probably would have been negative. Fortunately, I went and got tested again after about 9 days, and I was still negative. But I still stuck out the 14-day quarantine just to be absolutely safe and sure. So testing is vitally important, but when you get tested, can also make a big difference. So you can find out you're a close contact and get tested, and that test be negative, and it doesn't mean that the next test isn't going to be positive.
I would add to this that yes, I like the idea of frequent testing, especially if you need to be around other people. And part of the issue is the viral load that you have. So let's say someone gets exposed to someone else and actually gets infected. Well, 5 minutes later, if you do a test, the test is going to be negative. And it's very likely that that person is not infectious yet, that they're not going to be able to spread virus to someone else. So if I got a test, or we were going to have a meeting and everyone tested negative, if we have a meeting in the next hour or two, I think it's highly unlikely that someone is going to suddenly change. But a negative test tonight does not mean that you're not going to become infectious in a day or 2 or 3, so that it does off, the closer you are to the test, the more reassured I would be. The further out, just as Dr. Brissette mentioned, the more concerned I'd be. So I know institutions where they're testing the student body, this is on a college campus, twice a week. So they're doing it routinely, twice a week. I don't know if you can still hear me. It looks like, can you hear me? Okay. So, optimally, I would say twice a week. When I was asked about this for the North Dakota legislature, when they're planning their session come January, that's actually what I recommended if they have to hold in-person meetings, because that brings then the period down to a day or 2 in a regular 5-day week, when you'd be away for, where the incubation period might be there. That's probably about as safe as you can be. Once a week is pretty darn good, because it gives you some comfort level. But again, remember my paranoid statement from before. While we're in the midst of this pandemic, assume that anyone that you come in contact with is positive. And if you use that as your defense mechanism, it's about the best you can do. But yes, I like the idea of widespread testing once a week, particularly with one of those antigen tests that are much less expensive, may not be quite as sensitive, but enable you to test a lot of people frequently.
Dr. Brissette, did you have something else? I saw you quick raise your hand. Did you have something else you want to add?
Yeah, I just wanted to reiterate what Dr. Wynne was saying about assume that the person you're interacting with has COVID. And, to go back to my story with my student, we were both masked. I did not get sick. My child has been quarantined because he was notified that he was a close contact of another child at school who was not masked. My son has been wearing a mask. He's been negative. So just that simple barrier makes a huge, huge difference. And so if you put that precautionary piece in there, and if you're able to get, testing, the frequent washing of your hands, the keeping the distance, if you do all of those things, you can really protect yourself and everyone around you.
Great, thank you. The next question is, how soon do you think the saliva tests will be available over the counter in North Dakota?
I don't know.
Thank you. Okay. This one's kind of towards Dr. Brissette, but how stable are the test samples with that transport medium? And can they be stable without the transport medium?
Yes, absolutely. That's a great question. And I looked up some of the scientific literature on this before this meeting today. So the virus does survive on surfaces for a certain amount of time, and so it would survive on those swabs for a certain amount of time. But the ability of that virus to be intact and for us to be able to detect its genetic material really decreases over time. And so the recommendation is that you put the swab in the viral transport media, it helps stabilize it, and then you get that into the refrigerator and test within a couple of days, and you're probably, if there's virus on that sample, you'll get a positive result. But it's a function of both time and temperature. So the virus can be destroyed by temperature. If you think about the viral transport media, it's kind of like a broth that has salts and it has sort of the right pH so that it's more like your bodily fluids, and so it's a nice, it helps keep that viral particle intact. And so without that transport media, the virus could be degraded fairly quickly. The other thing is that the viruses, this virus seems to survive better on smooth surfaces and things that are rough. And if you think about the swabs, they're going to have that sort of, rough, cottony. So again, another reason to get it into that transport media and keep it stable.
Great, thank you. Okay, so when deaths are reported, we see that patients either had or did not have underlying conditions. Could you explain a little bit further why underlying conditions are particularly problematic if you're infected with SARS-CoV-2?
Well, this is actually a fairly ubiquitous finding. As a cardiologist, I take care of people who often have many other comorbid conditions, and it is just a widely observed finding that people who have other problems are just at particular risk. Now, there's certain ones where it's obvious as to why. So someone who has chronic lung disease and then gets a lung infection, there's no surprise there. There's some others that are theoretical where we're not sure, and I don't know if any of the other panelists might want to comment on this, but it turns out that there's a particular receptor related to this particular virus, which also is related to high blood pressure. So there was some thought that maybe people with high blood pressure and on a particular medication could be at increased risk or maybe decreased risk. And the bottom line is now that people are basically doing this. We're not sure, but probably just keep doing what you're doing. But we do know that people with high blood pressure are at increased risk. Now, is it because of this unique receptor that's involved? We don't know, but we do know that people who have diabetes, lung disease, high blood pressure, coronary disease, and especially obesity are at increased risk. Those are common observations, and the trouble is for a lot of those, you can't change it to reduce your risk, at least in the short term, with the virus. Same thing with age. I mean, I wish I could magically lose years too, but I can't do it. So the important thing then is for us as the public and as public health workers to protect the vulnerable populations. The trouble is we don't always know who is vulnerable. That's why herd immunity is a bad idea. But for instance, at the University of North Dakota, we are very aggressive about trying to accommodate anyone who is aware of some reason why they're at increased risk, whether it's age or comorbid conditions. And we really encourage those people to work with their supervisors so that we can lower the risk to them to the maximum extent possible. So I think that we don't know the exact explanation always in answer to the question, but we do know that that association is real, and we need to protect those people who are at increased risk.
And I'll just include also that to be clear that the protein that on the surface of the cell in the lungs that the virus binds to, this ACE2, the receptor for the virus that it binds to, is not only found in the lungs, it's found in your arteries, in your heart, in your kidney, in the intestines. And so it's a little early. We don't exactly sure the extent to which this virus infiltrates other parts of the body in addition to the obvious locations such as the lungs. So that could also be directly related or contribute to these other, the comorbidities that he was talking about.
Great, thank you.
Yeah, and I think something else with the comorbidities, and I'm just speculating here as someone who studies host pathogen interactions, but a lot of those diseases that Dr. Wynne mentioned heart disease, diabetes, they often have an inflammatory component. And so, if you think about your immune system trying to deal with this infection, if your immune system is kind of weak, maybe you can't knock that viral load down a lot in the beginning, and so you might have a worse outcome. But if your immune system is already sort of hyperactive and on the other end of the spectrum, you could also have sort of collateral damage. So, it's a balancing act. But I think one of the underlying components of a lot of these comorbidities is this kind of inflammatory process that's going on in people's bodies. So that may be another thing why certain comorbidities may make you more susceptible to bad outcomes with this virus.
Well, in support of what Dr. Brissette just mentioned, there was a study that was just published in the New England Journal of Medicine that if you use the anti-inflammatory agent colchicine, which is something that's used for gout, if you use that in people with coronary heart disease, they have better outcomes. So that again supports the idea that inflammation is could be playing an important role in all of this.
Great. Okay, so thinking ahead to future pandemics, what lessons do you think we have learned from the current COVID-19 pandemic, and how can we be resilient towards future pandemics?
So I would say there are a couple of important things that we've learned. One is the vital importance of a robust and well-developed public health system throughout the state and the country. Now, I think North Dakota profited by having a pretty darn good public health setup in both the state level and at the local public health units. That was not the case around the country. So certainly as a country, we need to invest more and better in the public health realm. The second is that it is shocking that the richest country on earth did not have adequate supplies of protective equipment, PPE, and so forth on hand and ready to go. On the other hand, there's certain things that we've done right, and the virologists can talk about this more, but we have never developed a vaccine in this amount of time, particularly for this type of bug, ever before. And that's because we built on a foundation that was robust and well established as far as our pharmaceutical industry, and that was based on a lot of research that's gone on over the years. So one of the lessons that we learned is how important that base is to develop novel approaches in a very rapid way. Some of the vaccines that have been developed have never been developed this way before, but they build on a platform that has been around for a while. So I think we did some things right, but I think better public health infrastructure, better equipment, and so forth. And also, one of the challenges that we have to do, I would say maybe the biggest challenge, the one that literally keeps me up at night, is how we reinstill a feeling in the American people that science and evidence and truth are paramount, and that we as citizens need to adjust our behavior in accordance with established facts. We don't have a randomized clinical trial that masks help, but I don't think there's been a randomized clinical trial that jumping out of an airplane without a parachute is not a good idea. So I don't think we need randomized clinical trials when there is a wealth of epidemiologic evidence that doing four things, I won't bore you again by going over them again, but doing four things is the principal way that we're going to turn this pandemic around.
And to add to that, as I tried to highlight in my earlier part of my discussion, is that we have to invest more attention in trying to identify the possibility of a pandemic event early to prevent it from spreading globally before we attack it. So we have to invest more in trying to stop the emergence at that stage one and helping to prevent, the best of our ability, to prevent spillover into the human population to begin with, which is a very challenging and difficult thing to do on a global scale because it involves so many components, everything from the government to business to illegal activities to surveillance of the animal populations. I mean, this is a very, very difficult, but right now it's almost as if it's kind of a niche academic research sort of, I'm exaggerating a little bit, but we're not investing enough. We've never been able to predict the onset of any pandemic thus far. We have to do better at trying to find ways in monitoring those driving forces that give rise to these in the first place, and try and control it at the very early stages before it gets spread all over the world. And then it becomes a whole nother problem, right? And then you have even socioeconomic issues associated with how you treat it. You have haves and have-nots. You have people who can control it and others that can't. So the whole can of worms opens up. And to try and stop it earlier, the earlier we can stop it, the better. And that's going to require an investment on a number of levels. And I think there's probably going to be more buy-in for the importance of this after we reflect on what we've gone through in the last year or so.
I mean, it really comes down to communication and coordination, and as Dr. Peliska talked about in his portion about all these different stakeholders coming together, and that's a really difficult thing to do, but we absolutely have to do it. And just an important thing about public health, people often feel like, well, there was all this hullabaloo and then nothing happened, and that's kind of the point with public health, is that if public health is doing a really good job, nothing is going to happen, and that is the point. And so I just want to reiterate the points that the other panelists have made, but really that communication and coordination and that investment in our public health infrastructure. And I say that as someone who got her degree at a school of public health.
Great, thank you. I think too, for me, it's encouraging to see how many scientists have come together in such a short period of time to work on something that hopefully we can continue with future pandemics, learning how to come together and work together for the better of the human race and the human population.
And it's quite remarkable, just to put this in perspective. Back in the when I started working in the area of HIV biochemistry in the late 80s, 90s, and 2000s, the speed at which new technologies, new methods, new ways of investigating the virology, the epidemiology of that pandemic was staggering at the time. I remember looking back and thinking, there's just nothing that has progressed. Our knowledge has progressed so at just a tremendous rate when scientists put their efforts into trying to understand HIV. And what we've seen in the last 8 or 9 months is dwarfing that, mostly because, as Dr. Wynne indicated, we're kind of using what we've learned from the past and the technologies and all the challenges we've came up against trying to develop vaccines against HIV. And all of these things have benefited, have helped us kind of be in a position to rapidly attack this problem, and I'm very thankful for that, and I hope it continues. It's the Pfizer announcement the other day was very encouraging and quite remarkable, to be quite honest. It's quite remarkable. It still waits to be seen, though. It's still a little bit early to count our chickens, right? And then we have to also have a discussion on what a potential endpoint of an efficacious vaccine means. What does it mean to have an effective vaccine? That can also have, different levels of meaning as well. We can discuss that perhaps if you want.
That actually leads to my next question. So could you elaborate on the implications of Pfizer's announcement with their vaccine showing 90% effectiveness in preventing COVID-19? And what are the next steps? And are there any estimates that we could have for a timeline of when we might see this widely distributed?
So the study is ongoing, and what they reported, just as you said, is that it was more than 90% effective. There are two critical things in a vaccine and also with other pharmaceuticals is obviously safety and efficacy. So those are the two things that the studies are ongoing to look at. Based on the official guidelines for approval and so forth, it seems like it's a reasonable estimate that sometime in December, if the ongoing trials continue to show what they're showing, that sometime in December or the end of the calendar year, that this could get approved for human use. One of the things that the government did that I think is brilliant, well, it's conceptually simple, but to have the willpower to do it, is this Operation Warp Speed. So what they did was they selected candidate vaccines before they were actually developed or even proven to be safe and effective, and started manufacturing them. So Pfizer has indicated that they may have something like 50 million doses available by the end of the year or early next year. Now, for the Pfizer vaccine, you need 2 doses. So whatever number of doses they say, divided by 2 for the number of people who can get vaccinated. But the drug companies started developing the vaccines and getting everything set up to manufacture them, even though they haven't been approved yet. What's the worst that happens is the virus doesn't work out and we're out $1 billion or whatever. Well, okay, $1 billion is a lot of money or whatever it is, but it's chump change if you think of the global economy and the potential impact that this will have. So Dr. Fauci has been quoted as saying that it could be late this year or early next year when people are getting vaccinated. Now, the people that the vaccine will be used on first are clearly healthcare workers and people who are at very highest risk. So for the rest of us who are not at high risk, it's not going to be immediately available. But based on the gearing up that these pharmaceutical companies are doing, it is a reasonable estimate to think that we're going to, if everything works out, and I agree, we don't know yet, let's not count our chickens before they hatch, but if things work out, thinking about a major change in the way life goes on in the second or third quarter of next year is a reasonable guess based on the way things are going. So, my plea to all of us is, let's not let our guard down, hang in there. Things are looking cautiously optimistic, and that we may be able to get on the other side of this sometime next year, so that Thanksgiving next year may look a whole lot different from Thanksgiving this year.
Pfizer themselves said that they projected they could produce up to 1.3 billion doses in 2021. But globally, even globally, that is clearly not enough globally. But here's what I take out of the early successful indications coming out of the Pfizer is that there's a number of other companies, one other company in particular that's in a phase 3 that's using the same messenger RNA technology to produce a vaccine. So we're going to get, and they're in the midst of it. So I think we're going to get pretty quick confirmation on whether or not this strategy is going to work. The particular strategy that Pfizer used. We'll get confirmation. And then there's, as Dr. Wynne indicated, there's a number of others, 5, 6 other companies who are in late stage phase 3 clinical studies. And the fact that we're getting good responses from one vaccine development bodes well for the potential of others developing also effective vaccines. So it's going to take, a number of companies with a number of vaccines to produce the magnitude of doses that we're going to need to treat this globally. But boy, 90%, what is measles? And is that like 98, 97, 98? If the 90 holds out, influenza is what, on the average 40, 50%. So, I mean, 90% is quite phenomenal for the first go round. So very encouraging, not only for Pfizer's direct results, but encouraging for the hope that the other vaccines will, find be useful as well.
So here's the arithmetic. There's something like 7.5 billion people on Earth. To get herd immunity for the Earth, you need to be 60 or 70% of that. So, you were talking about 4 to 5 billion people. So if one drug company can produce, what did you say, 1.3 billion doses?
Yeah, that's what Pfizer said they could do in 2021.
So that's 650 or 700 million people, and you have 4 or 5 or 6 drug companies that do it. I mean, you can start to see the orders of scale are possible. Final comment is there was on this issue at least, there was a recent very important paper that looked at humoral immunity, not the cells, but what occurs in your blood. And this looked at antibody levels in Iceland. And this study was done relatively early in the pandemic, and they looked at any number of antibodies. But what they found was that the majority of people, over 90% of people, had sustained and good levels of antibodies for the 4 months of the study. So we don't know anything beyond that, but there had been some talk that the immunity may go away very quickly. We don't know the answer to that yet, but this encouraging study suggested that a robust immunologic response lasted for at least 4 months in the great majority of people exposed to SARS-CoV-2. So that's encouraging that a vaccine may give us sustained protection. Hey, but look, if I need a booster once a year, I'm fine with that.
Yeah, and I'm going to give some optimistic sort of speculation here. This virus has a very large genome. And as such, that for it to have continued replication stability enough so that it doesn't mutate itself out of existence, it has actually what's called a proofreading ability when it's replicating itself, which means that this RNA genome, notoriously RNA viruses, are they undergo rapid evolution and generate drug resistance. I'm hopeful that the higher fidelity, apparent higher fidelity, at least in the studies that I've shown for the replication of this virus, might generate a more stable genome that will assist it. See, I told you I'm being speculative, but let's speculate here for a second, right? So we'll see, right? This is what started the development of a HIV vaccine, for example, is the rapid evolution and change of its genome due to the low fidelity of replication by the reverse transcriptase. So it's let's be hopeful, right? And the other thing that I want to say about the RNA virus, that Pfizer, it's not perfect by any means, because one of the technological challenges associated with these types of vaccines is that the vaccine needs to be, in their case, needs to be refrigerated at minus 80 degrees centigrade, which technologically is not a problem at UND, or even for small doses at the University of Mary, but worldwide that poses a technological challenge that has to be overcome. How do you store this magnitude of doses at such a low temperature until they're ready to use? So I just wanted to point that out as well, that there's other technological issues and practical issues that have to be overcome for this to work out with this particular vaccine.
Great. Okay, I have one last quick question before we close out. This is more of a general question. So what would you tell a high school or college student that's interested in pursuing a career similar to yours?
Go for it. I can't believe I've been as lucky as I have been to be able to combine taking care of patients, doing science, teaching, and through my administrative roles, trying to make the world a better place. I mean, I don't know, I can't imagine anything more exciting. My only regret, I guess, is that I don't do more of the fundamental science that some of my colleagues on the panel do. I haven't done that for many years, but this is such a marvelous career. I would say go for it. If you have an interest, go do it.
So my advice to students is always try different stuff out. You may think that you really want to be a physician and you wanted to do that your entire life. And then maybe you get into a research lab and realize that you really love doing the science part. Of course, you can combine those two things. You can do both. But sometimes you just don't know until you try it out. So if you have a general interest in biology or nature or medicine, seek out different experiences, try different things. And my other advice is that I was in a hurry to get to wherever I was going. And I wish I had taken some time after my undergraduate and not immediately gone to graduate school, because I wasn't absolutely sure what I wanted to do with the rest of my life. And it is absolutely okay if you're a young person and you're not sure what you want to do with the rest of your life. It's okay to go out and work. If you're in a position you can take a gap year, if you want to join the military and travel the world or do the Peace Corps, something. I love seeing applications come in when I look at graduate students or medical students and see people with some life experience. It's okay not to be in a hurry. It feels like a really long road and you think, oh my gosh, I'm going to be in school until I'm 30. Trust me, it's just not that long, and you can always change what you're doing. My dad got his PhD 3 days before I graduated from high school. So you can always go back to school. You can always change your path. So don't be in too much of a hurry.
I agree 100% with that. I've been a pre-medicine advisor for undergraduates for over 20 years now, and an academic advisor for countless students. And I've seen it all. I've seen students who come in saying they want to be a physician and leave being a research scientist or a dentist or a chiropractor. I see students coming in convinced that they want to go to graduate school only to go to medical school. I've seen it all. And I agree, find out what you're good at, find out what brings you joy and gets you excited, and go for it. Don't fall in, and I agree with you exactly that. In my own travels, I kind of did the next obvious thing. And I said, okay, I'm done with my undergraduate. What's the next thing to do? Oh, you go get your PhD. Okay, you go on, you get your PhD. What's the next thing you do? Oh, go to postdoc. Okay, I went to do a postdoc. And then what do you do after a postdoc? Well, you either go into industry. I went into academics. So without ever stopping in between and saying, okay, this is what I can do and what's typical, but is it what I should do and is it what I really want to do? In the end, it turned out great for me. I'm very pleased and happy with where I am, but I think that perhaps I would have changed some things along the way had I kind of contemplated a little bit more about what the next step was. And perhaps, you know, maybe I would have taken a different road. I don't know. But yeah, slow down, you know, contemplate. Think of it not as just a job, but a life vocation, something that you really involves your life that is a big part. We spend so much of our time doing this, right? So much of our life doing this. Find something that brings you joy and brings you, you know, a sense of accomplishment and go with that, right? And find good people along the way to help you, right? And they're there. Sometimes they're not always obvious, but, you know, seek them out. Seek out those experiences, like you said, and talk to people, you know, and you'll come out just great, right? And keep plugging, right? Keep plugging away, work hard.
Awesome. Thank you, guys. Well, I think with that, we'll go ahead and wrap up. Thank you all for a wonderful discussion. And I would like our audience members to please join me in virtually thanking our panelists. In the UND School of Medicine newsletter, Dr. Wynne mentions that although we should maintain physically distanced, we don't have to stay socially distanced. And so I'm very thankful for opportunities such as this one, that we can engage in a virtual discussion so we can still be socially active while maintaining our physical distance. So thank you to Dr. Peliska, Dr. Brissette, and Dr. Wynne for taking time to share your insights with us tonight. And I would like to thank Gateway to Science for hosting the event. And the Visiting Scientist Series was honored to feature these regional scientists and the great work that they are doing. Please, I invite you all to check out the Visiting Scientist Series Facebook page, as well as gatewaytoscience.org/VSS for any upcoming presentations. Thank you all for joining us, and I hope you all have a wonderful evening.